Hey, we'll go ahead and try to knock as much of this as we out as we can in just a few short minutes. So we're going to talk about the composition of blood really quickly. Now, blood is made up of a few different things. It's made of plasma, um, and it's also made of formed elements. So, plasma takes up about 55% of our total blood volume. And of that 55%, about 90% is water. But that plasma also contains things such as proteins, electrolytes, nutrients, such as glucose, amino acids, and lipids, as well as hormones, and waste products as well. Our formed elements are things like red blood cells, white blood cells, as well as platelets, and these take up about 45% of our total volume of blood. Um, yeah. As red blood cells go throughout the blood, the blood, they have about a life cycle of 120 days. They should be shaped like this over here on the left, but after about 120 days, they become a weird shape. They become more rounded or oval shaped, and these rounded shapes actually get filtered out through the spleen um, and broken down inside the spleen. So the spleen will come in and it will break down our red blood cells into hema, um, iron, as well as amino acids. Now, these, oh man, I should have taken notes on that. The iron and amino acids get sent right back into the blood and they're recycled. They're made back into red blood cells via our bone marrow. But our hema is converted into biliverdin, which is polar, and it is then changed into bilirubin, which is nonpolar, and this all occurs via enzymes within the blood. So all these at this step are exited outside of the spleen and they're into the bloodstream now. So our hema is breaking down by enzymes into biliverdin, which is polar. Um, it is then it is then made into a nonpolar molecule, which is fix that really quickly. It only does that whenever I'm recording videos. But then it is by an enzyme turned into a bilirubin from biliverdin, at which point it goes inside the liver, and the liver adds glucuronic acid. This makes that bilirubin a polar bilirubin, which then gets interacted on by bile as well as so the bile gets into it and it interacts with it, and then that bile gets taken into our intestines, which at which point the bilirubin will be eaten by bacteria and will be turned into urobilinogen. Now, if that urobilinogen is it has two routes that it can go into the intestines, at which point it'll be a stereobilia, which is brown, which makes up our poop, or it can go into our kidneys, which in which case it becomes urobilin, which is yellow, which is the substance that we see in our urine when we're particularly dehydrated. Um, awesome. Describe the three mechanisms that contribute to hemostasis, vasoconstriction, platelet aggregation, and coagulation. Awesome. That brings us down here to hemostasis and coagulation. So this comes becomes very important when something. Oh, fascinating. Yeah. So whenever the hemostasis and clotting ultimately helps us to repair our vessels and keep ourselves from bleeding too much. Now, when our capillaries get damaged and the cell membrane is broken, or excuse me, the capillary membrane is broken, as seen here. Um, uh, Obviously, we start to bleed. Our body doesn't want us to bleed too much, and so what it'll do is it'll cause our sphincter, our arterial sphincter, to uh, um, not dilate, but to constrict, uh, making it so that less blood can flow into there, ultimately making it so that we lose less blood to that cut. And that's what vasoconstriction helps out with. The next step of our reducing blood and repairing the repairing the membrane is or the capillary lining is maybe a better way to say that, is all along the capillary wall here. We have epithelial cells, as seen by these little ridges right here. These epithelial cells line the whole of the capillary walls, and they release what's called VWF, which stands for von Willebrand. This VWF is particularly affinant to collagen fibers that are exposed whenever a cut happens inside of our capillaries. So what happens is these VWFs from over here go all the way over to here to our cut site and end up binding with the blue collagen right here. And that's all based off charges. I knew that was about to happen. Awesome. Oof, it does not like when I stream. So those VWFs will bind to our collagen here, and these VWFs bind to it and on one end, and they leave their other end open. Now we have platelets, which are have what are called GP1B receptors. Now, these are VWF receptors. So one side of those VWFs will be on the collagen, and the other side will be exposed. And these platelets will come in and they will bind their receptor to the exposed end of that VWF. Once it starts binding, that the platelet, as seen here, turns into a weird looking little octopus thing. Basically, it expands and grows, and it ends up releasing. It ends up releasing a few different um, particles here. It releases thrombonin, I believe is what that's called, a TXA2, as well as dense granules and alpha granules. Alpha granules we won't care too much about with our current knowledge, but dense granules and TXA2 are very important to know. Dense granules bind to GP2B3A with a fibrinogen um, due to ADP release. Um, uh, so the ATP gets released and it will bind to both platelets, thromboxic. It'll bind to platelets as well as TXA2 receptors to help them facilitate clotting quicker. Let's see. On the ADP, I said those receptors. Okay, yeah. The ADP also binds to fibrinogen receptors that are found on these platelets, as indicated by the red here. So whenever our platelet becomes this weird octopus thing, it sends out those chemicals and it ends up recruiting other platelets to the cause to stop the bleeding. These platelets will come around the central platelet, and they'll already have started their conformation changes due to the receptors being filled. And they'll get over here, and fibrinogen, as seen here in the blue, will bind fibrinogen receptors between the platelets to form a webbing of sorts, a loose webbing, very loose webbing. But it'll connect all of these, and that is done through the GP2B, 3A sites. So ADP and fibrinogen will bind to these sites, allowing them to bind together. Okay. And then we have our clotting factors here. So whenever um, the blue part, goodness, I can't remember what it's called. Whenever the collagen becomes exposed, we have factors within our blood that uh, will interact with the collagen and eventually cause it to begin to turn fibrinogen into fibrin. So we have intrinsic pathways as well as extrinsic pathways associated with this. Intrinsic is referring to the factors within the blood interacting with the collagen directly outside of the capillary. And extrinsic would be if we got a deeper cut and these factors interacted with muscle tissue. Uh, so muscle tissue and other cells, other tissues. But intrinsic is a little bit slower of the method of helping or aiding in clots. Extrinsic is a little bit quicker. Then we also have the common pathway. Both extrinsic and intrinsic utilize the common pathway in their final stages of clotting. So what happens here is a 12A factor, or excuse me, a 12 factor, or factor 12 comes in and interacts with the collagen because collagen has a negative charge with it, and it activates that factor 12. A factor 11 will come along and interact with that activated factor 12 and form an activated factor 11. Factor 9 will come along and interact with that factor 11A and will form a factor 9A. At this point, a cofactor 8 will come in and interact with our 9A, and that will lead us to a factor that will create factor 10. This factor 10 will interact with factor 9A and cofactor 8, and this will form factor 10A, which when acted or interacted with factor 5, cofactor 5, will turn factor 2 into factor 2A, also known as thrombonin. This thrombonin can turn factor 1, which is fibrinogen, into factor 1A, which is known as fibrin strands. So basically, this whole process here turns fibrinogen into fibrin strands. That loose web I was referring to earlier over here with the blue lines for the fibrinogen will become fibrin, which are thin strands that are much tighter bound. It'll form a much tighter web and prepare it to be contracted. Okay. So there we covered our intrinsic and common pathways. Let's quickly go over our extrinsic pathways. So with our extrinsic, like I was saying, we interact with tissues. Now these tissues have a factor 3, yes, factor 3, also known as tissue factor, bound to these tissues. So we have a factor 7 that comes in and interacts with those tissue factors, turning it into a factor 7A. At which point factor 10 can come along and interact with 7A, and that'll turn it into a 10A, so a 10 activated. And then we just go over here and continue on with our common pathway with a 10 interacting with, with 2 interacting with factor 10A and factor 5 to create factor 2A, also known as thrombonin. And then factor 1 will interact, fibrinogen will interact with 2A to form our fibrin strands. So both of these, extrinsic and intrinsic, both lead to the production of fibrinogen. Now, what's interesting is that they both of these, so because they're two different processes, can be blocked by two different drugs. With extrinsic, if we use warfarin or comactin, this blocks vitamin K, which is essential for our factor 7 to interact with our tissue factors. And so when we block that vitamin K with warfarin, then we prevent the extrinsic process from occurring. With intrinsic, when we use heparin, this blocks the intrinsic pathway and makes it so that we can't clot through that pathway. Um, for intrinsic, we use the PTT test to test for the clotting there, and we use extrinsic PT to test for the extrinsic clotting. Okay, intrinsic. Man, plasminogen. That's what we need to go over. Okay. So TPA, plasminogen, and plasmin all work together in the breaking down of these clots or scabs. Um, so TPA is short for tissue plasminogen activator, which is released by endothelial cells. It converts plasminogen, an inactive protein within the plasma, into plasmin. This plasmin is an enzyme that breaks down fibrin, effectively dissolving the clot in a process called fibrinolysis. Um, so yeah, that whole process, it just makes it so that uh, the clot is broken down once the lining of the capillary is repaired. And paste that. Sweet. And that concludes the video. Thank you for watching.